Good morning, everyone. Thanks for showing up so early. Really, I appreciate that. So I'm going to start the session, which is about um, the ways how we can manipulate and measure neural oscillations using brain stimulation tools combined with neuroimaging by focusing on EEG and MEG and how we can use those tools to study neural oscillations specifically. So why should we do that in the first place? I think because oscillations are ubiquitous in the brain. They're a very widespread phenomenon, and they're involved in many different cognitive functions. And they're also aberrant in a lot of neurological and psychiatric disease. And I think they are so widely found in the brain because they are reflecting basic computational principles. And the most basic one might be rhythmic input-output gain modulation. That means no matter how exactly an oscillation is generated, it usually has a more excitable and a less excitable phase. Let's just call it the upstate and the downstate. And during this more excitable upstate, the likelihood to have a neuron population fire when there's input is increased, while during the downstate, it's less likely that the signal is transducted when input is coming in because those neurons might be hyperpolarized or actively inhibited even. And this gives rise to a number of other principles, like communication through coherence. The idea that if you have overlapping duty cycles when information processing is occurring, so if neuron populations are in sync, then brain regions or neuron populations can talk to each other, while if they're out of sync, they cannot. And there's cross-frequency phase amplitude coupling, because during the more excitable phase of a slower oscillation, there is an increase in amplitude in faster oscillations, and in the more excitable phase of those oscillations, again, even faster oscillations are grouped. And thereby we get a hierarchical nesting of oscillations and more fine-grained temporal resolution throughout the brain. Another principle that's affected by now this phase amplitude coupling is, for example, the idea of pulse inhibition, which has been proposed for alpha oscillations. So there's this idea that alpha represents an inhibitory asymmetric oscillation with bouts of inhibition rhythmically suppressing gamma oscillations, which reflect bottom-up sensory processing. So with increasing amplitude, you would have increasingly shorter duty cycles and less time for gamma oscillations to occur. And this would work to route information flow in the brain by inhibiting task-irrelevant brain regions, increasing alpha, and by disinhibiting the task-relevant brain regions. And this might be under control of the prefrontal cortex, for example, the frontal eye fields. And finally, there's also the idea that phase amplitude coupling might subserve synaptic plasticity in a way that high-frequency input due to the high-frequency oscillation coinciding with a more depolarized phase, for example, in this case of a slow oscillation, for slow oscillation in spindles being coupled. This might provide the optimal scenario to get very strong calcium influx into the apical dendrites of neurons and create LTP-like plasticity. So just showing a few of these general principles that have been proposed, there might be a good reason to believe that these oscillations are very important states of the brain and we should consider them. And we can study them, of course, with different neuroimaging modalities, which come with a different spatial and temporal resolution. And EEG and MEG seem to be the most applicable non-invasive tools to study neural oscillations of these frequencies. And you can combine different modalities even, but it all remains correlational until you actually manipulate brain activity using, for example, transcranial magnetic stimulation or transcranial direct and alternating current stimulation, so TCS. And they come with a similar spatial and temporal resolution, and we can combine them also with those imaging modalities. And you're going to hear today examples of TMS EEG, TCS MEG, TCS fMRI, and I'm going to focus on TMS EEG and TCS MEG today. So, in principle, what you can do when combining these approaches to study brain oscillations, you can go for an online or an offline approach. Online means you image or record while you stimulate, and you look for the direct response of the brain to the stimulation. And offline would mean that you use a protocol to induce synaptic plasticity, and then you look for the subsequent after effects that arise from these synaptic changes. And based on the protocol, you can induce some LTP-like facilitation or LTD-like depression of cortical excitability. And also the online approaches come in 3 different flavors. You can quantify brain activity, stimulating networks strong enough to provide a direct output. You stimulate the motor cortex to get this motor evoked potential, the MEP, or for other brain regions you would get at least a TMS evoked EEG potential. You can interfere with task-related but also spontaneously ongoing activity in the brain, or you can just more gently modulate it using, for example, rhythmic TMS or TSES to modulate the timing or to entrain, to use a strong word, neuronal activity. So going back to this idea of pulse inhibition, this is something we try to investigate by combining online TSES with MEG. So the idea is that we try to mimic the alpha oscillation by stimulating at individual alpha frequency, the visual cortex, and we started a few seconds before the onset of an inward moving high contrast grating, which is typically producing a strong gamma response in the visual cortex. And now the idea was that we have difficulties removing the TSES artifact from EEG and from MEG, but we can look at higher frequencies and how the gamma power is modulated by TSES here. So how gamma is modulated by a low frequency oscillation. And so we stimulated with individual alpha frequency, but also with 2 flanker frequencies, minus and plus 4 Hertz, to see how specific this response would be. And importantly, because we know that TSES can produce phosphines of retinal origin. So because the retina is more excitable than the cortex for this technique, we also used a frontal control montage, both of those montages being titrated individually to 80% phosphine threshold, so they wouldn't see any phosphines, but we also wouldn't get any subliminar retinothalamo-cortical entrainment, because what we're looking for is transcranial entrainment. And all these trials were intermingled within a session. And we had some sham trials in between, and for those sham stimulation, you see the very typical gamma response to the visual stimulus lasting as long as the stimulus lasts. While when stimulating with occipital montage in the upper row, you see as a difference a strong reduction in gamma power, while for the frontal control montage, that was not the case. So it seems to be that the gamma response to the visual stimulus is suppressed indeed by the TSES, or the occipital cortex, and more so it is phasically. So if you look at the distribution of power throughout the TSES cycle, we see that at the frequency band where the visual induced gamma response happens, we see a phasic modulation, and it seems to be a phasic suppression. So we also quantify that, and we see indeed a stronger phase amplitude coupling for the occipital compared to the frontal montage, and also compared to surrogate data. But it didn't differ across frequencies. So it doesn't look like we're entraining alpha, but more, well, entraining neural activity per se, maybe just hijacking the membrane potential, shifting it forth and back at the stimulation frequency. So entrainment in a weaker sense. We also find a weak but significant behavioral effect in a way that the participants had to indicate the rotation direction of a very small asterisk in the middle of the stimulus, and they became worse in doing so during the occipital stimulation compared to the frontal one. And the stronger TSES modulated the visual induced gamma response, the stronger was this behavioral impairment. So it seems to be behaviorally relevant. And it looks like this pulsed inhibition is actually what is happening, or what we can mimic with also TSES of the visual cortex. But now let's leave the visual cortex and go to the sensory-motor cortex, because also there is an alpha oscillation. It's called the sensory-motor mu alpha. And here we use a different technique to see whether there's also pulse inhibition or maybe something else. So the classical approach would treat the brain as a black box, right? Just stimulate the brain no matter what's going on in the brain at that time. And we get some output measure with a large variance. But if we would then sort according to the brain state, so for example, the oscillatory power or phase of a certain frequency band the brain was in at the time of stimulation, we might get very different responses, each of them being more homogeneous. And if we would know that brain state at the time of stimulation, we could target specific states. And this is what I would call brain state-dependent brain stimulation. And this is what we used here in the study. So we detected the brains that we were interested in this case with EEG, made some decisions based on power and phase, and then stimulated. It's not a fully closed loop here, so we are just characterizing certain brain states, but we're not modulating them. And the idea is to separate these three scenarios, so the mu alpha being asymmetric pulse inhibition, as I've shown before, maybe excitation and inhibition being modulated, or whether it's asymmetric pulsed facilitation actually. And we did so by targeting two different states of alpha power, the upper 20% of the individual mu power distribution or the lower 20%. The upper 20% would be strongly expressed mu rhythm, and the lower 20% would be strong desynchronization, almost an absence of this oscillation. And in that case, we could also only target random phase, but for the high amplitude, we targeted 4 different phase angles to see whether there would be phasic modulation of excitability. And we used single pulse TMS to just measure the MEP, so we stimulate the motor cortex, and looking at the MEP amplitude as a measure of cortical spinal excitability, and we used also paired pulses to assess GABA-A-ergic inhibition by the short intercortical inhibition protocol. Now, and this is just to show that the targeting went fine, and on the left side you do not see an alpha peak. This is for the random stimulation during the low alpha condition, while then for the high alpha condition you see a clear alpha peak in all the cases. The blue and red lines are just for the single and paired pulses. And here you see time frequency representation. It ends on the right side at time point 0 when we give the TMS pulse, so there's an increase in the alpha band in a few hundred milliseconds before we apply the stimulation. So don't get confused with the blue and red. It's Z normalized across all the conditions. And what we do also see is, same case here, that the alpha power is actually originating from the stimulation side, because we use a local Hjorth montage, a local Laplacian, to extract the signal from the left sensory-motor region. And it's not confounded with some spill-in from the occipital cortex here. But most importantly, we see that we target the right phase, so the upstate, or let's say the positive peak here, the falling flank, the negative peak or trough, and the rising flank. So we used EEG trigger TMS to target these different brain states and assess them, and let's come to the results. So compared to the left, in yellow here, the low power random stimulation, we see that there's no change during high power trials if you're at the positive peak, which is less excitable. But if you're at the negative peak, so the trough, which is more excitable, then you see a difference compared to the other three conditions. So what we do have is, if there's a strong alpha oscillation, there is a pulsed facilitation during the troughs. There's no pulse inhibition, not in the motor cortex. We also do not see any modulation of the short intercortical inhibition. It is definitely there for each of those states, but it's not modulated. And now, with the second smaller study, we also looked at whether then there should be also a positive relationship between the power in principle and the excitability, which is what you would predict from the results on the left. And this is indeed what we find. It's a weak, but it's a positive relationship. And this is well in line with what Hartwig Siebner showed yesterday, and also what Leo Cohn's group has found. So in the motor cortex, cortical excitability seems to be positively related to the power of the sensory-motor mu rhythm. Why is that? We don't know yet. It is possible that due to the strong inhibitory connections between the sensory and the somatosensory in the motor cortex, this is in fact a pulsed disinhibition by a pulse inhibition of this somatosensory cortex. But future work would need to identify that. So no matter how it's actually working, there's, let's say, at least first evidence that if you can repeatedly now target one of the states, for example, only the troughs, that you get a facilitation of the cortical excitability that lasts at least half an hour. Or if you target the same power state, but only the less excitable peaks, you do not get this kind of effect. And this is well in line with the mechanism I proposed, that having a high frequency input during a disinhibited or facilitated phase of the oscillation will produce synaptic plasticity. And so where's this going to lead? In the future, hopefully, we might be able to close the loop to actually stimulate not only to characterize brain activity, but to modulate it, to use really ongoing brain signals and brain stimulation to close the loop. This is at the moment very difficult due to the artifacts we induce with the stimulation. And just as my last slide, I promise to advertise this toolbox which is published. It's an open source MATLAB toolbox for the external control of TMS devices. This is what comes in handy if you want to change certain parameters like stimulation intensities, interstimulus intervals, pulse form, and so forth online during an experiment, maybe doing a closed loop stimulation. So I would like to close here and thank my collaborators from Tübingen and elsewhere, and thank you very much for actually coming in during the talk and getting a decent crowd. Thank you. Thank you very much.